Uh, I think I see Mohit also. Mohit, you'll be sharing your slides, right?
Yeah, hi. Hello, everyone. Yes,
Hi.
I'll be sharing my screen.
Okay, so Sri Gauri goes first, then Rahul, and then Mohit. And then we have the panel discussion with Praveen and Mitali, and that will be moderated by Srishti.
Yes.
So
And
I'll
the
be,
Q&A.
sorry? Yeah, Q&A at the end, yeah. So I will be introducing uh, both Premas and Omics logic in the beginning, and then panelists and the speakers for the day in the introduction itself. So before each talk, I will not be introducing again. I will just say over to you, Mohit, or over to you, Rahul. Okay. I hope that is okay.
Yeah, perfectly fine.
All right, Arushi, let the attendees in.
Yeah.
Karishma, you and Arushi also right now in Bangalore?
No,
Yeah.
I'm in Pune, Praveen.
I'm in
Arushi is
Oh,
in Bangalore. Yes.
okay. Are you based
India,
in Pune?
yes, I'm based. I live and work out of Pune.
Oh, wonderful, wonderful.
Yeah.
Hello, everyone. Good afternoon. We have almost 35 people already in. It's just increasing. Welcome, everybody. So we'll just start in a minute. We'll just let everybody join in. Okay, it is 3 o'clock. So according to India Biosign Standard Time, we should begin the webinar on time. So hello everyone and good afternoon. Thank you all for joining us today. My name is Arushi and I hope you all are doing well. On behalf of India Bioscience and Premas Life Sciences, I'm pleased to welcome you all to this interactive and informative webinar. The topic that we are going to be covering for today's session is the IIT Jodhpur certification program on next generation omics technologies and applications for 2024 and 2025. This is a course that has been collaboratively developed by Premas Life Sciences and Omics Logic. So before I go about introducing them to you all and the speakers and the panelists for the day, I would like to do a quick technical check. So on your Zoom panel, under the reactions tab, there must be a raise hand icon. Can you all just click on this once to indicate that you can hear me and see the first slide that is visible? Okay, I see a few hands raised. So I'm assuming that everything is correct. Okay, great. Thank you so much. Just a little bit of housekeeping before we get started. Your mics will be muted during the session. So if you have any questions, please type them into the Q&A box and we'll be taking them up post the presentations. So today's session, we'll be focusing on understanding this program and how to go about you all submitting your applications. So the applications are open for the IIT Jodhpur certification program now, and I think the last date is July 25th. It's a 6-month online program, and it provides participants with introductory skills and knowledge necessary to design, execute, and analyze multi-omics experiments. So you will be knowing about the program in detail through our speakers and panelists for the day. But before I introduce the speakers and panelists, I would like to give you a brief introduction about Premas Life Sciences and Omics Logic. So Premas Life Sciences is a young, dynamic, and focused organization introducing game-changing niche technologies in genomics, cell biology, and biopharma to boost innovative research and diagnostics in India. And they're also the knowledge partners to several reputed research institutes and hospitals, enabling them to set up core genomics facilities with complete support at all fronts. Omics Logic was formerly known as Spine Biotech. It's a US-based company, a leading innovator in NGS bioinformatics, specializing in training programs developed for analyzing such high-throughput omics data. So for the panelists for the day, we have Mitali Mukerji, who is the professor and head of Department of Bioscience and Bioengineering, IIT Jodhpur. She was formerly a chief scientist at CSIR IGIP, which is also my alma mater, and was involved in setting up key genomics initiatives in the area of rare genetic disorders, population genomics, functional genomics of repetitive sequences, and genomics. So we welcome you, Dr. Mitali. Praveen Gupta is the managing director at Premas Life Sciences and has over 23 years of experience in life science and molecular diagnostic arena, handling solutions for cell biology, proteomics, high throughput screening, and genomics. We also have Mohit and Rahul. Mohit is the Chief Executive Officer at Omics Logic and is also the program advisor to this PG diploma program on next generation omics and technologies applications. Rahul is also a training and development specialist at Premas Life Sciences and is also a program mentor for the same. So we welcome all the panelists for the day. I would now like to introduce our in-house experts and speakers. Srishti is the Assistant Marketing Manager at Premas Life Sciences and will also be our moderator for the panel discussion with Praveen and Mitali today. Sri Gauri is the Scientific Communications Manager at Omics Logic, and she'll be introducing the course for today. So without further ado, I'll hand over the stage to Sri Gauri, and Sri Gauri, you can start.
Thank you so much. I hope my audio is perfectly fine.
Yes, Sri Gauri.
All right, perfect. So hello everyone, I'm Sri Gauri, and in my part of the presentation, I'll be introducing you to the postgraduate diploma program, the various features of the program, and a brief overview into the program curriculum and the various opportunities that are available and a quick registration process overview. But we'll be covering and exploring each of these topics much more in depth during our upcoming session with our expert team. And if you have any questions, you can discuss it at the open floor session towards the end of the webinar. So as Arushi initially explained, the postgraduate diploma program, it is a comprehensive training program that has been designed for participants to equip them with the necessary skills and knowledge to design, execute, and analyze multi-omics experiments. And in the program, we cover a diverse range of topics, including next-generation sequencing techniques, multi-omics technologies, computational methods, machine learning algorithms, and statistical approaches. And for the ease, the training program is divided into 3 flexible program options. So we have the basic certificate program, which is for a period of 6 months, and advanced certificate program for a period of 6 months as well, and the combined postgraduate diploma program for a period of 12 months. Now, who is this program for? The individuals who enroll for the program must have a bachelor's degree in engineering or science or medicine, or even a master's degree. And we also look forward to participants who are working professionals from various sectors, including pharma, sports, nutrition, health, wellness, and diagnostic industries, researchers from academia and industry, clinicians and service providers, as well as PhD students. Now, this is the team of experts that we have during our program, the directors, mentors, and advisors. And as you can see, we have an expert team from a diverse range from the academia as well as from the industry. We have faculties from the IIT Jodhpur and Ashoka University and experts from Premas Life Sciences and Omics Logic as well. Now, just to give you a quick overview into the relevance of this field in today's market, but this topic will be much more better explained by the team of experts during the panel's discussion. But just for your quick overview, as you can see here, we have the stats on the left that explains the growth of this market from 2024 to 2030, which is about 18.6%. And regional-wise outlook, it is more prominent in North America, Europe, Asia Pacific, Latin America, Middle East, and Africa as well. And the application-wise outlook, we are also covering a few of these topics in our program as well, such as genomics, molecular phylogenetics, metabolomics, proteomics, transcriptomics, and other fields. Now, this is the program overview of the program curriculum that we have. There are 2 aspects to it, compulsory modules that the students are required to complete, as well as the elective modules. So we'll be explaining much more in depth in the upcoming session, but here is a quick overview of these modules. We have comprehensively explained this in our program brochure as well. We'll share the link in the chat box so that interested participants can dive deep into it. Now, one of the unique highlight of this program is that the students get the opportunity to collaborate with either an IIT Jodhpur faculty member or industry partner to apply their learning throughout the program into a practical research question. And here we have provided some of the recent student projects that have been carried out, and they can be carried out either as a mini project in a group setting or as an independent mini project. So just to give you a quick overview into some of these excellent projects that have been carried out by Anamta and Sonal, who worked on identifying the biomarkers in psoriasis patients, and another group project that has been carried out on bacteria whole genome sequencing by a team of students Arvind and Sakara. Manisha, who worked on developing a machine learning model for feature extraction of breast cancer subtypes, and Mrinal, who worked on developing an R package for extracting and visualizing genomic metadata from the NCBI. And finally, another interesting project that has been worked on as a group by Sita and Rajesh on transcriptomic analysis of long COVID patients. Now, the other important highlight is that the students also get the opportunity for campus immersion, where students will be provided hands-on learning experience during the on-site IIT Jodhpur laboratory sessions involving techniques related to Omics technologies. So here we have provided some of the photos from our recent campus immersion, where students worked on sample and library preparation for bacterial whole genome sequencing. So the students benefit not only from the practical sessions, but as well as from the in-person lectures and discussions that are held at the IIT Jodhpur, as well as the opportunity to network with the industry partners who will also be present in the campus immersion phase. Now, coming to the program outcomes. So at the end of the program, students will have in-depth knowledge of the high-throughput multi-omic technologies, and they will be able to perform next-generation sequencing experiments and interpret their applications. They will be able to learn the computational tools and statistical concept to analyze omic data and make meaningful biological interpretation. They will also develop skills for project planning and management in omics and data science. And one of the important outcomes is that this curriculum, it has been designed by a team of academia and industry experts, which are in alignment with the latest trends and practices. Students will also benefit from this curriculum as well. Now, here's a quick roadmap of how this program has been structured in a quick overview manner. So when you submit the applications, it will undergo a process of review, and if you're selected, you will receive an offer letter, and you will be progressing to the certification 1. Now, at the end of the certification 1, you have the opportunity to exit with the certificate 1, and we also provide participants a direct entry at certificate 2. Now, who are these participants? These participants must have completed similar courses as in certificate 1, either through prior formal academic trainings. And also, to qualify for the PG diploma at the end, these participants should also complete 13 additional credits as electives. Now, most often we are also encountered with the questions like, what are the various opportunities? Again, we'll be exploring much more in depth in the upcoming sections by as well as in the panel discussion. But just to give you a quick overview, we have the opportunities in wet lab as well as dry lab, and as displayed on the screen, we have opportunities in academia, industry, as well as internships, and as well as the dry lab opportunities in academia, industry, and internships as well. So this is just a quick overview. It has been comprehensively given in the program brochure as well. So interested participants can take a look at it. Now, who are the potential recruiters if one is interested in this field? So as you can see, the industry partners, they are from a diverse range of sectors, from pharmaceutical research institutions, healthcare organizations, data analytics, and genomic diagnostic laboratories as well. And here's a quick feedback of the student. Overall, students have given a good feedback about completing the coursework, as well as the course recommendation and the difficulty of the coursework has been given. And these are a few of the glimpses into students sharing their experience during the recent campus immersion on LinkedIn. So we also regularly repost all of this on our handle, so participants can take a look at it if they are interested. And students also share their feedback. And as you can see, they are from a diverse range of backgrounds, from biochemistry, pharmacy, and biotechnology. They share their experience on how they plan to proceed ahead. Now, coming to the program details, just a quick overview. We have covered most of the duration and credits part in the previous slides. But just to give you a quick overview, the credits is 13 for Certificate 1, 15 for Certificate 2, and 28 for PG Diploma program. And the live sessions that they get opportunity to interact with the mentor, it is approximately 196 hours in the Certificate 1, 252 hours in Certificate 2, and approximately 448 hours in PG Diploma program. And the program pricing is 1.2 lakhs for Certificate 1, 1.5 lakhs for Certificate 2, and 2.5 lakhs for PG Diploma program. Now, this is a quick overview of the registration process, the final slide for those participants who are interested to register. So the first step is to submit your application in the registration form. We'll share the link in the chat box. If you have not submitted your interest yet, do fill out the form and you will receive the next steps. That is to submit your registration fee of rupees 300. And then you will be required to share your documents for verification for eligibility into the program. If you're deemed eligible, you will receive an offer letter for the next steps. And you will be required to make the complete program fee payment, and then the program will commence from August of 2024. So I just wanted to quickly wrap up as my time is there. I've noticed it in the chat box. So that is all that I wanted to cover in my part of the slides. And next we have a wonderful presentation from Dr. Rahul Ramekar, who will be explaining more about the omics experiments and technologies aspect. So over to you, sir.
Hello, everybody. Just give me one moment. I'll share my screen.
Yeah,
So
sure,
can you
Rahul.
let me know if the slides are okay.
Yeah, we can see your screen and everything works fine.
Yeah, so this is fine. Okay, fine. Thank you. Thank you very much. So thank you, Shri Gauri, for briefing about the PG Diploma course. So one thing, have you noticed that when she was discussing about the topics, so we heard a lot about omics. So what are omics? So when we are so here, as you likely know that the central dogma of biology, it details how information is encoded in an organism and it eventually becomes phenotype. So here what happens is the crucial information is encoded in a cell's genes, and that is transcribed into RNA, which is then translated into protein. So here, of course, the regulation of how DNA is transcribed is often controlled by, we can say, non-genetic regulatory mechanisms, such as epigenomic or epigenetic regulation. Here, when we're talking about omics, so each layer of the central dogma, the genome, the epigenome, the transcriptome, and the proteome are considered as omes. And omics is an umbrella, we can say umbrella term, referring to the study of individual or multiple levels of central dogma. So, So now, when we are talking about genomics or when we are talking about genome, it is the complete description of one's genetic makeup. And we can say the variation in the genes defines the phenotype that can underline many diseases or different traits we can discuss. So such variation could either be germline variation or somatic variation. And studying these kind of variation with different approaches such as whole genome sequencing, whole exome sequencing, or targeted sequencing can come under genomics. So whereas when we are talking about transcriptomics. So here, not all of the changes to the genome are due to changes in base pairings. So what you can say that the way in which DNA is organized or chemically modified also significantly tells us the story of gene influencing phenotypes. Yeah. So this is about epigenomics, what I'm talking about. So here, the cumulative term studying for non-genetic changes is called epigenomics. So on the other hand, RNAs are the functional output of one gene. So here, all cells, we can say that have the same DNA, yet the gene expression varies drastically between different cell types. And studying these gene expression comes under transcriptomics. So at the last layer or level, we can say, is the proteome. So it is the oome that is closest to the phenotype. It is the functional output of all other levels of cellular regulation, we can say, including the genome, the epigenome, and transcriptome. And this is where researchers often look when they're interested in finding things like biomarkers. So, like, the question comes that why do we need to study omics? So if we omics or multiomics, so if we take cancer research as an example, here each ohm tells us something distinct story about cancer biology. If you're looking at the genome, it often provides the mutational profiles that help to predict patient diagnostic, patient prognosis, or it can also classify tumors into meaningful groups, or even we can say diagnose cancers. So whereas when you're considering the transcriptome here, since it is the functional output of one's, we can say, gene, it can show us pathways that are up, or we can say down regulating in specific cancer. Whereas the epigenome, it adds an explanatory, we can say, mechanistic dimension to RNA expression. Here, it can further refine tumor classification. And finally, the proteome, which is the closest to the cellular phenotype, it can provide biomarkers and other phenotypic information that responds to all, or we can say, predict the therapeutic responses. So this is how we should integrate our further research using omics. But like all other technologies, so even we can say omics is also plagued with different challenges. So what are the challenges? So biological complexities is one thing. There are ethical and privacy concerns. There is a rapid evolution or in technology, because technology is dynamic, right? There are wet lab challenges, and these are the challenges we need to address. But how can we address them? So we can address them by reshaping the skill-based approach. So this is what actually we are doing now by this PG diploma course. So we are designing a training program that will help to build a skilled workforce which is proficient in omics technologies, what we have discussed. It can narrow down the training and expertise gap. It can provide skill sets to efficiently plan multi-omics experiments. Yeah, but the thing what actually we believe is, so whatever thing the theory what we have learned, so based on that we can say that the true development of skill comes from hands-on training, where practical application turns the theoretical knowledge into real-world expertise. And this is what we believe when we are delivering the PG diploma course to you all. So whatever theory we have learned or you will learn in the course, we will do a hands-on training. So we will try to apply with the real-world samples. And this is what actually we did in campus immersion a while back. So, and actually with this, what I'll do, I'll hand over to Dr. Mohit, where he will brief you about the data analytics part of the course. Thank you.
Good afternoon, everyone. Thank you, Dr. Rahul and the team. It's a pleasure speaking with you today about the fascinating world of omics data science. Nowadays, generating omics data is a common activity. That is what we are learning and hearing all the time. I was recently reading this, and that I read that Kerala turns to genomic sequencing for dengue, sickle cell anemia, and treatments. It was in a national paper, and you'd see such news more often than not in the mainstream news, and which is a very, very good thing for all of us here, because this has been a need of the hour. So what we have learned is that, and many of you know, that genomics, transcriptomics, proteomics, metabolomics are arguably the most developed and used omics. But there are other omics studies as well, like all of these omics studies actually describes the functioning of the cell, which requires integrate relationships at different omics level. For example, epigenomics describes the transitionary chemical modification of the DNA, and lipidomics looks at the lipidic subpopulation of metabolites. So omics diversity mirrors the complexity of cell systems. So with the constant improvements of measuring techniques such as next generation sequencing, possibility to access ever larger subsystems of cells are increasing. So omics dataset generation and pairing it with software development, essential tools and technologies that are involving led to the omics data science field, which is an interdisciplinary field that kind of integrates various omics technologies um, to understand the roles, relationships, and the action of the molecules of the cell of an organism. Uh, in this PG diploma program, we will be utilizing a lot of commercial as well as open source tools. And to give you an overview, at Omics Logic, what we have done is that we have prepared a pipeline for for everyone to start from very beginner level, which means that, as you have seen, that many students that we get are from different background, educational background, coming from different type of expertise that they have. To personalize it and to make sure that everyone is at the same level, we start from there, and then we deep dive into different type of omics technology, their application using machine learning and data science. So the omics logic is going to be fully utilized in this course, which means that all that you see here will be available for all the students and students who will be working with us throughout this year as the postgraduate diploma program or the certificate 1 and certificate 2. So the training modules are placed like this, where you will be able to learn from a lot of such resources, as you can see on my screen. All of this is placed together in a curriculum manner, so that it all makes sense and nobody is missing out on anything and getting overwhelmed. So learning from the beginner level to intermediate level, and then trying to work on an advanced level. So how do we do this in a very unique fashion is because of the hands-on part and the project-based learning. So once you learn about the technology, the next thing is to learn about the application of that technology for a specific type of research. And that sort of research is being simplified and provided to you as courses that you'll be able to learn from very easily. And there will be a mentor to support all of this throughout this period. So this is a very unique program at IIT Jodhpur that we're really proud of, because I think I haven't found anything of such a program across anywhere in the world. And we are an international organization where we are integrating this into different type of uh, curriculums, where you will not be able to use just the resources that I saw, but also everything in one place, learning about all of this in much detail with the help of help of a mentor, and then to be able to translate it into research or research interest or a project. That is what we have learned. So this is the entire workflow, where you start from a beginner level, then you move into technologies, learn about data science, learn about omics data science, and learn about the different steps in data science. For example, data processing, data integration, combining data from different omics platforms. Data processing means cleaning and normalization. So there is a lot of statistics which is going to be involved in this. And I think when we think about data, actually, in any respect, we think about visualization, that how that data would look. We think about graphs. We think about statistics. I just couldn't fail to notice that we just finished a cricket World Cup, and even in those times I see so much statistics coming into the screens, making everyone interesting. Similar things happen in bioinformatics. When you combine data, when you integrate data, then you see different types of pattern, and those patterns lead to discovery. And to do this, we have different types of tools: statistical analysis, machine learning, and AI. We talked about, biomarkers. There is personalized medicine, there is drug discovery, there is agriculture, there is environmental science, and also sports and health, which are like one of the major areas in this. So, as I said, this is a very unique opportunity where industry is participating along with best of academia and best of industry. And there we are combining all of this into a unique experience. And given the opportunity that we have to work with you for 6 months or 1 year, makes it really, really possible. So, in conclusion, I'll just say that I'm looking forward for all of you to learn more about this, and maybe join us in this program and get recognition that one would deserve after learning so much and trying to, apply this. So all of this is available here for you to review, and we have great outcomes coming from the first few batches of the students, and we look forward to learning more from you. Thank you so much.
Thank you so much, Mohit, Rahul, and Shri Gauri, all three of you. You people handled the moderation as well when you spoke on your own. So thank you so much. And now we have Srishti, who's going to be the moderator for the panel discussion with Praveen and Mitali. So over to you, Srishti.
Thank you, Arushi. Thank you for the introduction, and thank you, Dr. Rahul and Dr. Mohit, for introducing the course, and also, the importance of omics and the data integration part. So we look forward to the course. So we have eminent speakers and panelists with us today, Dr. Mitali and Praveen, and we would like to have them on board and discuss about the importance of multi-omics. So my first question would be for Dr. Mitali. So Dr. Mitali, if you could please briefly explain about omics and what do it refers to and its significance in the modern research nowadays.
Uh, so the omics is the study of the entire genetic, meaning the attributes of the entire genetic makeup of an individual. It can be any life form. It could be a virus like the COVID. It could be the human. So how they behave, how they are different, how do you know what is inherited? Any functional attribute which we see in an organism is an outcome of our genetic makeup, and how do we study them is all the technologies around it, and which is now become multi-omics technologies. So you start from DNA, it will be genomics, then you look at its expression, it becomes transcriptomics. At the RNA level, expression at the protein level becomes proteomics, lipid level becomes lipidomics, metabolite level becomes metabolomics. So you study the entire thing in entirety. So you need technologies. Earlier we would do one thing at a time. Now there are platform technologies where you can do all of it together. So that would be the aspect. And then all life forms, be it bacteria or be it human, we all have the same language, DNA, same AGCT. So as a result of which, same set of technologies can be used for all these diverse things. And there's also extensive sharing, so that you can understand meaning of one life form from studying the other life form. So it opens up a whole gamut of possibilities for understanding life on Earth. So as a result of which, if you study any from any perspective, you can apply it to different applications. So I think that's in a nutshell. I would like to say that this is all omics.
So thank you, Dr. Mitali, for briefly introducing about omics, its importance, and also shedding some light on the technical aspects of it. We would like to have pose another question for our panelist, Praveen. So we would like to know that, could you provide some insights on how the dynamics of genomics have influenced research in India over the past decade?
Okay. Thank you, Srishti. Thank you, India Biosciences. We are sitting with one of the pioneers in the genomics field. Professor Mitali is here. I still remember those days, ma'am. If you remember, 20 years back, you were working with IGVDB, trying to catalog and create the India's first, database for cataloging all the variations um, from across the different populations. And I still remember how complex as a project, and it's a multi-centered project with so many collaborators. I forgot how many authors were there in that paper, but I think something 150 plus.
200.
200.
200.
Imagine. So a massive project of that scale was undertaken about 20 years back to create to start the initiation of genomics at a high scale. Of course, we've been doing genomics for many years before too. Professor S.K. Brahmachari is a pioneer in that area. But I think this particular project led to kind of looking at high throughputness or scale, or looking at large sample sizes and looking at large complex data to be cataloged in the country for the first time. From there till now, today we are talking about, recently DBT announced the completion of Genome India, and we've done 10,000 genomes now, whole genome sequencing. This is at a very high depth coverage of cataloging every single base pair of humans, Indian population, covering about 70 plus population types or subtypes in the country. So I see that the way genomics has probably influenced is that whether it's human, whether it's plants, whether it's animals, whether it's bacteria, whether it's viruses, it's greatly influenced the scale and the speed and in unraveling the complexities of different omics per se. I'll give you one example, a very common basic application during COVID. The way India took up, the sequencing project to basically look at creating, cataloging the variations of all the new upcoming strains, and I think that became a household, I would say, exercise where I think a common man understood the importance of genomics, the importance of sequencing. And today, I think people understand what is variance. So that is, I think, so to me, I think it has come a long way, and I'm very excited to see where it is going. And as Dr. Mitali said, I think multi-omics is the way now, where people are looking at further complexities to combine, overlay data to see how we can understand biology even better and being more closer to the function per se.
Thank you, Praveen, for answering that question, because we understand, the importance of genomics and how we have come and progressed in the last 10 years, and now multi-omics is the future that we all need to look out for. Moving on to the next question, I would like to ask Dr. Mitali that, what are the challenges do we face in understanding the complexities of, the complex diseases, and also how the omics can help understand those complexities?
Complex diseases by nature are complex, right? It's not only genes which play a role, but it also is the environment, and how does the environment impact the expression of the genes, so that the same genetic makeup, maybe between related individuals, exhibit differences in the outcome. So you might have siblings having an equal chance of a predisposition, or maybe having a genetic makeup which is nearly similar, but they progress differently. And if you take this to the population, or to even different countries, then you will find that there is no set rules, it seems to be. And that's because the genome is very big. It's 3 billion bases, the human genome. And there are so many combinatorial possibilities, because there is a huge amount of variation. So between two unrelated individuals, we would vary at a million places. Now, if you think that a fraction of these variations can actually impact the functioning of our genes, or overall functioning, then how many combinatorial possibilities can come out of it? And then how do you kind of have some kind of a prediction as to who are differently predisposed? That is what we want to know, because then we can actually prevent diseases in some people. Why do some people interact, have differences with respect to medicines? or outcome, or what is the likely prognosis. So you want to keep on predicting from a person's genetic makeup, how likely is that person to be susceptible to disease, to have a adverse outcome if they have the disease, or how they could modulate the environment, and which environment they should modulate so that their genetic behavior is kept at optimum, right? So for all these things, you need multi-omic technologies, because you cannot, everything works in networks, and it could be the same network, but players might be different in different individuals, and so you need to study them in totality. So that's the reason that in complex diseases, multi-omic technologies have become like the bread and butter of trying to tackle it. So large number of success stories have come out. It's not that everything is known now, but we are starting to build these this knowledge up, and as the knowledge grows, and this is so high throughput, and people are making the data available, it also gives us an opportunity to do meta analysis and not do all the experiments ourselves, and port information. And that's where the computational part of the omics comes in, very, very important. So this is what our course caters to. There are advancements in the technologies. So if you just develop the skills, then you might be just limited to doing that much. But what is the foundational knowledge you should have? So as the technology increases, or as the data increases, how do you tackle that, and how you become relevant at all spaces, is what this course is all about.
Yeah, I mean, we completely agree with you that, we can have multi-omics resolve so many complex diseases and have like a big, huge database which we can refer to, and also the importance of computational biology and bioinformatics. And the course that we all are offering helps us understand the importance and the relevance of, multi-omics. Praveen, you've mentioned about the importance and how pandemic has made us realize that the importance of genomics and multi-omics. But then there's another question that I would like to ask you is that that currently, what is the current status of the omics research in India, and where all we as compared to the global landscape, where India stands?
Okay. um, so, I mean, my simple answer would be that we've done a lot, but there's a lot to be done. So I think with now the advances in technology available, we were talking about the whole advancements per se. So with the advent of technologies like NGS, and now new technologies like proximity extension assays, where people are trying to combine to study even protein or proteome at a large scale using the same genomics machines. With the availability of technologies, the way the world is adopting these technologies into large scale projects is at a different scale altogether. You look at UK Biobank, they've already done 500,000 sample profiling, for whole genome sequencing. And now, based on the data which is coming out, they've actually made certain guidelines in the NHS system that now every cancer patient or every rare disorder patient gets a whole genome sequencing in the clinical practice per se. So, and like this, there are zillion other projects. India too has done a lot of projects. We've had some, so IGVDB was one, but I mean, we have Genome India, we have Indigen, we have ICGC, we have ICGA, which is going on right now. We are looking at so many mycobacterium genome sequencing large scale projects which are being done right now. So at every scale, the work has been going on. But I still believe, looking at the large population size of the country, we need to do more. And I think we need to do more multi-omic studies now. At small scale, a lot of labs are now beginning to realize to combine this layering of omic data and not just looking at one dimension per se. So as Dr. Mitali and Rahul explained, that how people were earlier looking at either whole genome sequencing and maybe at the most transcriptome level. But now, if you can overlay with looking at large number of proteins together, there are about 2 lakh proteins, say in human side. So normally, when you're studying maybe 30, 40, 50 proteins together, instead of that, if you're looking at studying about 5,000, 6,000, 10,000 proteins together at one go, and if technology is available, that is where I think those adoptions have to start in the country. Some labs are doing it, but I think it's still at a very low scale. I would love to see more of that happening.
Yeah, I mean, we all of us here in the panelists and also the audience would agree that, there's a lot of scope of growing the multiomics domain. We have small labs doing experiments, but there's a lot of scope of expanding the multiomics domain and having a lot of sample inputs as well. Talking about the another challenge that currently we are facing is that, the clinical applications of multiomics and how we can, help and face or solve those challenges that we are currently facing in terms of, the omics research and their applications in the clinical settings. Dr. Mitali, if you could answer that.
So, I think, like Praveen mentioned, our contribution in the genomic space is comparatively very, very low. As Meaning, if we think about our population size, our diversity, the number of patient pool which we have more participated in international projects by giving our samples to outside for analysis than really doing that much work in India in a collaborative setup. Genome India was the first biggest one, and also the ICGA. So there is an enormous scope, and I think what we need to do is we need to build an ecosystem of every stakeholder, and amongst them, the clinician become one very, very core important component, because the clinicians have a better understanding of a disease, and they have an handle to the phenotypes. But the genomic people in the labs have more knowledge about maybe what happens to the if to ascribe functionality. But if we do not make both of them crosstalk, then the right kind of an outcome what a clinician might at the end need for an application to be easily translatable into a clinical space might not be so easily done. So as a result of which, it now we need to have at every space hybrid people, doctors who know a little bit of genomics to have a good conversation with genomics people, genomic people to have a conversation with clinicians to be able to tune their analysis to much better way, computational people to be able to have the ability to talk with the doctors for an appropriate problem to develop their skill sets. So this is all very, very complementary need and complementary expertise which we need to gain. So what we have in in this diploma program, or whatever we have developed, it's in that philosophy we have developed this, that if we have this thing imparted to the students, they could have variable trajectory. It could be clinician coming into it and having a knowledge so that they can also plan better experiments. If they plan better experiment, then the service providers or the company people like Praveen will be able to get better reagents across to them, which are very, very useful. The genomics will also grow because the moment clinicians have started to acknowledge that this is the need, they'll start analyzing it. So it's a very needed area. Or at this point of time, what is most limiting is doctors think they have the samples, they can give it to a service provider does a routine analysis. Populations are different, cases are different, India is very different from Western world, all the standards are based on European population. The same diet is not there, the same environment is not there, the same exposures are not there. So we need to really, there's a major need for all these annotations. So in the clinical space, there's a huge amount of need and a huge amount of opportunity to integrate omics, and it's not only for the clinicians, but it is also for people who are going to do genomics.
Thank you, Dr. Mitali, for for making us understand that it is important for us to bridge the gap between the government as well as the private sector, so that we have stakeholders and a strong built ecosystem. Which brings to me, which brings to the last question that I would like to ask Praveen is that. Since already Dr. Mitali has explained about having an ecosystem, how can the public and private partnership strengthens our field, and specifically how might academic institutions like IIT Jodhpur is also doing, we can have collaborations done to build this ecosystem and have training programs in India specifically.
Thanks for that question, Shrishti. And I think it is paramount, So I think if you look at if you look at the global scale, if you look at US, which is very advanced right now, the whole advancement of science or the whole application of science or the whole translation of science occurs when there's a equal public-private partnerships. These are formal, informal ways of basically tying up. So as they say, see, the skill set first comes in the academic space because they are basic scientists who are looking at science at a very deeper level. They have a functional understanding of the technology. They have a functional understanding of the application area. So it's very important that tomorrow, if we can forge ways of communication between industry and academia, and again, there are various ways on a smaller scale. We can have small cohorts of meetings, to meet together and understanding what are the challenges which industry has and which academic institute has expertise to solve that problem. And tomorrow, if an approach requires, like as I said, biology is complex. Diagnostic needs to get more deeper to basically stratify a patient for a personalized genomics approach. Today, people are using genomics approaches. Tomorrow, people will be using genome, epigenome, proteome, microbiome, exosomes, and the list can continue on. Now, as the data, as there are more layers of data which is coming out and the complexity is increasing, obviously the skill set in the industry requires hand holding by the academia to basically work together so that, as Dr. Mitali said, meaningful deductions are done. Otherwise, we might just probably make deductions based on maybe very isolated markers, which may or may not be very comprehensive, or we are just trying to copy paste Caucasian data into our context and then basically making inferences. So to me, I think it's paramount that first of all, an open mindset between both academia and industry should be there to forge, partnerships. There are many ideas. I think I would love to see in India when a large-scale government project has a both public and private component where, assuming the next large genomes, project. Suppose India does 100,000 genome project. How industry and academia work together to complete that project because the capacity is available at both the sides. So I think those kinds of initiative, if they can be done with all the checks and balances, put together for data security and stuff like that, that will, I think, pave way for a lot of industry academia collaborations.
Thank you, Praveen. Thank you, Professor Mitali, for answering and shedding lights on the importance of multi-omics and how we can establish an ecosystem and bring such collaborations further with other partners as well. So yeah, that's all from my side, Arushi. I think we can take questions from the participants.
Yes. And we already have a lot of questions. Some I think Shubhangana has already answered. So thanks, Shubhangana. But I'll start. So I will still request till I'm taking the questions from the Q&A box. If people have more questions, keep typing them in. We still have close to about 20-25 minutes. So we will try to answer most of your questions. So Manisha Pandey asks, what is the difference between certificate 1 and certificate 2? So anybody, Shri Gauri, Rahul, Mohit, or Shrishti, anybody?
Can I just
Yes, yes.
Yeah, so the certificate 1 is primarily foundational in nature, and it is kept open so that people are able to judge where they more gravitate towards. So you are given a little bit of genomics, a little bit of orientation to the computational aspect, and not little, but the courses are in depth, but the aspect is all foundational. It also will allow people to appreciate what is omics and appreciate to know that there is more to it, and if they want to develop further into an application specialist or want to get into computational genomics or want to get into more experimental, how the samples are prepared, etc. So the second one is more on hands on. So you do mini project, you do immersion, and you do whatever you learned in the foundational, plus you are exposed to huge number of experts also who are doing lots of genomics. You get an exposure to interact with them all those things. But I think the people, Mohit and all, can add further. I just wanted to give the broad philosophy on the basis of which we develop this two certificate. It's so, and there's also a break, so you can come back next year to do the certificate, but you at least go with an IIT, meaning a certificate. But then you have a IIT alumni. When if you do a diploma, then you become an IIT Jodhpur alumni, both the certificates together. So it's a stackable program.
Thank you, Dr. Mitali. Anybody would like to add on to this, or should we go to the next? Mohit, do you want to add on?
Yeah, I mean, actually, ma'am described all of this, but just to say that Certificate 1 is a foundational course and covers all the basics. And we have had 90% of the students opting for Certificate 2 after Certificate 1. So I think that is how it is well connected and gives a holistic understanding of, and from skills to application, I think it covers all of those things. So it is a great opportunity.
Okay. Another question from Manisha is, how tough is the course for someone from non-coding or non-mathematical background? I think she means that if somebody does not have a computational biology background or coding, how tough is it?
Okay, if I can, ma'am,
Yeah,
do you
yeah,
want to go?
or anybody.
Mohit, you go, then I can just add my bit to it.
Sure. So actually, we started off with this. The platform we started was for biologists and clinicians because we saw the gap that we discussed today, which is what they've been like, because they are the people who are doing the experiments and they did not come from from a coding background. So we developed this entire courses and training to help them. So we cover all of that in a very basic manner where we are learning about from installation to practicing codes, and there is a mentor to guide you all the time. That is a great help. So I think it is we have a lot of students, as you could see, most of them are from non-coding and non-mathematical background, but we're able to do it after the certification.
So I would like to add that genomics, today's day, you can complete all your experiments in 5 days time. Once you have the reagents, once you have the sample, you put it in a machine, it gives you, and you take a year to analyze it because half of us in the country do not have a foundational knowledge to even open the files. So so we have actually an introduction to R, introduction to packages, and these packages have well formed modules. So if you want to do some analysis and if you practice, you learn the packages, then you will be able to pretty much do basic analysis, and that's what the first certificate allows you to do. So if there is a paper you're reading in genomics, you can take the supplementary and you will be able to do a similar kind of an analysis using these packages. There's a fear how to do it, and this is what we want you to get out of. That's the skill part. And the next one is more of, if you were to tomorrow want to do your own experiment, want to contribute to an R&D lab, want to do anything which requires a little bit of thinking, then you get into application of all those learnings into projects and immersion programs. So I am not a computational biologist, but I have done 20 years of genomics. So you realize it, but my half of my students are computational biologists, and they know what to do with the data, and they really have the skills. So it's not very difficult. Most of them come from biology background.
Yeah.
I also want to add that I have also done my PhD with no computational background. So yes, you can do it. It's not very difficult.
Yeah, it's just basically you get time to get comfortable. Once you come,
Yeah,
and that's what we have observed in education all the time, that as ma'am said, like many people are not just ready to open it and see it, but once you get comfortable, everyone is capable of learning this.
yeah. So there's another question from an attendee who has asked, if we have to opt for diploma, do we have to pay for previous both certifications? can it be done completely on online basis? I think there are multiple questions. Maybe we take one by one. So
Mohit, you answer these.
Yes, so certificate one is 1.2 lakhs, and then if you want to go to certificate two after certificate one, it is 1.5 lakhs. But if you go for diploma, it's one time 2.5 lakhs. So either you're going for certificate one or you're going for the full program. So you can choose for the full program, so you make some savings out of it, and also get an opportunity to completely plan your studies accordingly. And there are exams in between, but it's built for professionals. So the classes will be over the weekend, so that you all have the time to, dedicate some hours for this course.
And loan possibilities are also there.
okay,
Yes.
that is nice. Any placements will be provided by the organization?
So we have developed a system about it. We are connecting the students with the right kind of opportunity, which is again personalized, because everyone has different skill sets that they come up with, which are their basic skill sets, and then they learn different types of skill sets and application. And then there are different types of choices, like the area where I want to do a job, also the specific field that I want to get into. So we have a small module about it, which helps students to customize that first, and then we help them connect with the right kind of opportunities when there. That is how we are helping
And
students
just
with
yeah, just
opportunity.
to add to what Dr. Mohit has said, so we are also trying internships. So
Hmm.
we are discussing with different companies how the students can get internships, paid internships. In addition to that, we are also helping them to get recommendations for PhDs. So how they can apply. So we are helping them how they can they prepare the cover letters, the CVs. So it's all personalized. So in this way also we are helping the candidates.
Praveen, you can
So,
also tell them about the
yeah.
opportunity space.
Yes, yes. I was about to come to that. Thank you, Dr. Tari. So, the omics space has a huge potential right now. I'll just give you a small, figure right now. So, right now, currently, it's estimated that globally there are about 30,000, say, NGS machines installed of different companies and makes and types, which are there right now. With all the pundits, all the business pundits, basically are saying that in next 5 years' time, it will be at least 100,000 instruments, if not more. Now look at if it increases from 30,000 to 100,000 installs globally, that means there's 70,000 new boxes coming up. On an average, for each box, you require about 5 or 6 manpower. So you can do the math. Approximately 4.5 lakh new jobs will be created in this space. That's one global data. Now let's come back to India. From a scope perspective, what's happening right now is there's a massive uptake of these tools right now, omics tool in the research area, but there's a even faster uptake of omics tools right now in the clinical area. So today we were doing an estimate right now. So approximately in India there are about 1.5 lakh pathology labs which are operating in different cities. I mean, every tier city, we have a pathology lab. Currently only maybe about 150 of them, in fact. And I mean, if I give you a more accurate figure, before COVID, the labs doing molecular diagnostic were not even, I mean, whether any kind of molecular diagnostic, whether it's Sanger, whether it's PCR, whether it's qPCR, it was hardly about 150, 200 labs in the country doing some kind of molecular diagnostics, and about 50 labs were doing sequencing. In last 4 5 years alone, now from those 50, now there are about 200 labs doing sequencing in the clinical space. And imagine, still we have 150,000 labs in this country. So as these technologies are becoming simpler, the cost is coming down, the data output is increasing, and the availability of manpower, skilled manpower, is coming in the market. The adoption rate of these technologies in path labs, hospitals, pharma companies, food testing companies is just spiraling. So we are very interesting. We can give you specific examples of how interesting job opportunities are coming up in this sector. And to me, I think it's a very exciting phase for anybody in the omics area. Provided you are skilled well, you understand the complexity pretty well, and you can actually analyze the data.
Yeah. Thank you, Praveen, Mitali,
And I just would like to mention that no master's programs are available which caters to training in this area. So
Yeah.
this is like a really an upskilling if you want to increase your job opportunities after masters. This diploma, if you consider the number of hours which we spend on real teaching, because this teaching, the course is very rigorous. It has gone through multiple iterations of the Senate before it, because it was being offered by IIT. So it's very rigorous, and it's internationally, it can, all of you, if you go through this course well, then you would be able to compete in equivalent space even internationally. So that's the thing I'm confident about. So
Yeah, I'm sure about that. So there are few more questions by the same attendee. That person asks, if a person can opt for online session, then will they be available in the recorded version as well? I think probably if they miss a class or something.
yeah, we give all the recordings. So these classes are typically held in Google Classroom.
Okay.
It's an environment which IIT, so you become a student with an IIT email, and all the notes, all the recordings, they are all provided in the Google Classroom. But we encourage people to attend, and we've made it such that all the people can attend on the weekends the classes. And but all the recordings and the all the teachers who are teaching, they are from company or IIT faculties, and even faculties from outside Ashoka and all. So we have a very interesting group of people who have had experience in genomics teaching, and they give personal time also sometimes.
So, some of you have already mentioned that the classes will be over weekends. So, some people have also asked what are the timings of these sessions, and will there be any exams that will be held during the course?
So there's continuous evaluation, and it runs from the timetable, because you can understand the number of hours you have to cover in in 6 months, right? So it is, weekends are pretty much tied up. Mohit, will you be able to add?
Yes,
Rahul?
actually, yes, we have theoretical sessions and then hands-on sessions equally divided. So it is always interesting, and you're learning from
So I will just answer that. So weekends are packed. So it starts from morning 8:30, and then we have the sessions, each session by instructors. And then, like ma'am has said, there are also recorded sessions, but we encourage you to join online so that you can get feedbacks, you can ask questions, so we can solve that immediately. Yeah, but it starts from morning 8:30. There will be some break time, and it will go until evening. Yeah.
Okay. There are certain questions related to omics, like what are some of the current wet lab challenges for omics out of genomics, epigenomics, proteomics, metabolomics, which is the most industrially relevant omics?
All are relevant depending on which industry you're going to and which question you're posing. So that's what this program actually tells you. Unfortunately, what happens is most of the people start with exome sequencing. That's the
Mm.
easiest thing to do, right? And you think that that is the universal answer for everything. That's only 1% of the genome. There is so many things. If you want to, there are many stakeholders in omics. I will just show you one slide. Maybe I just had it. Can I show
Yeah, yeah, sure.
different stakeholder? Yeah, let me show you one slide. Uh, it's a mind map actually, which I also teach during
I
the
have
course.
uh, I have seen this map in my course.
Okay, so you see, there are multi-omic technologies. You need to know the technology around each of them, and then you have the understand them in the context of a problem space. Integrate them: systems biology, ecosystem, exposure and omics, multi-omics and digital. So omics gets added to the next level: radiology, to ecology, to exposure, to multiomics. So all of them, and then there's a component of AI in there. So predictive models, all those I think are there. And then it leads to different kinds of research questions, maybe functional omics, precision medicine. Nowadays, there's a very important area of landscape genomics where you want to do biodiversity conservation. Then One Health, you want to look at everything in a holistic perspective. From So you need to sequence maybe all the organisms or some readouts. Drug target discovery, synthetic biology, agriculture, sociogenomics, how much is the society and genes interact. And then there are translational areas where algorithms and platform needs to be developed, and other drug discovery, digital health, sustainable health, biodiversity. So these are the places where one are going into entrepreneurship, and they need it. So there are different kinds of stakeholders in this. Um, just 1 minute. These stakeholders will be in the disease health and wellness group. These will be in public health, strategic sector, pharma, nutrition, and sports is kind of taking up a whole lot of things in omics. And then more importantly, in the industry, omics service providers, typically if they do service for huge number of hospitals and all, they need people, field application specialists like Praveen's huge base of sequencers all across the country, forensic labs, R&D labs, then precision agriculture, this is very important. Then ecology, or the people who are into landscape, biodiversity conservation, surveillance. And then the biomanufacturing, this is again another new upcoming area which is coming up in the country, where you need synthetic biology, lots of genomics is needed. So this is what I just wanted to show, that once you start from here, you can go into any area, because the DNA is universal, so it feeds onto all the areas. So that's where I wanted to show this mind map, just to say what are the opportunity spaces. So it could be research, industry, and translation.
Can I just add on something, ma'am? On this very, very interesting mind map, you covered practically all the applications. Let me just give you a couple of examples, very interesting examples, which have come to us in recent times, where people have basically bought our NGS machines for very interesting application. One, a food testing company, a very, I'll blind the name, a very, very large, very, very popular company. They make a lot of supplements, health supplements. So they're using NGS machines to basically find out that when they're selecting the constituents, suppose they're selecting a certain herbal constituent, they would like to use the genomics approach to find out whether there's a contamination, because they're buying it from certain sources. So they would like to use genomic stratification or genomic genotyping per se, in this case using NGS, to find out whether they're using pure material or not. That's a very interesting application. Recently, about 6 months back, a lab in, we never knew about this lab, a lab in Bangalore bought our mid throughput system, and we were very intrigued because we had never heard of this lab and we wanted to understand more. And when we actually went there, we understood. Now, this is a clinical service lab, which is only catering to the cosmetic industry, mainly in France. So what they're doing is, in the French cosmetic industry right now, whenever any cosmetic major player, all your Chanel's and everybody, when they make any cosmetic right now and it's to be applied on the skin, they have to do studies like before and after, what's the changes in microbiome. So this particular service provider is basically catering to the cosmetic industry just to study the changes in microbiome in terms of. So like this, there are very, I'll give you a very common example. I think most of will understand. Beer industry, it's a very large industry. All the European beer manufacturers today, because they use yeast as a fermenting agent. Now, if tomorrow there's a mutation in the yeast stock, then it basically changes the taste of the beer. So they use NGS or genomics or omics technologies to sequence before inoculation into every large batch. So like this, I can go on and on. There are so many applications coming up, very interesting applications coming up of omics per se. So it's a great field, and one needs this expertise to be, available for this workforce.
Very, very interesting applications. I also didn't know about a few. So Yeah, so there are a few more questions. I think Kanakana, I think your question is very specific. I would request you to please send an email to one of the panelists. Some people have asked what roles did the previous batch students go into. I think we've covered that, but they also want to specifically know what were their CTCs. So I'm not sure if you're okay, but any range that you would like to specify from anybody from the panelists or speakers.
So the first batch was very diverse. Some of them already are in jobs. Somebody wants to pursue for PhD. So many are up because this was the first time they had no idea what they were into. So they were teacher in college from pharma background. So this allowed them to understand how they can use omics in their space. There was a person who was in education, then company, then I think he's gone into education. One is coming working in a company. So many of them are employed already and they would be looking for more options, and many are wanting to pursue higher studies abroad, so they thought that they will get skilled over here. So it was a small batch because we just started it, and anything new in the country you introduce, everybody waits to see what happens to them, right? And I think one or two are approaching Rahul companies, right? So
Yes, yes, ma'am.
that's also happening. So this was just the first batch, and so we had few students. We are hoping that through this, because now we are more confident about our classes, our delivery, and we've been able to successfully do our immersion. So we have done one round, and the first round is the toughest round. But I think next round, people will also, the ones who are going to be the job providers, will also be looking forward to the kind of trainings that are being proposed. So
Yeah. Another question is, I think we don't have a lot of time left, so we'll probably take one or two questions. If somebody's question is not answered, I will please request them to, email the Omics Logic or Premas Life Sciences, and somebody will reply back to your email. But let me take a few more questions. What kind of computer requirements does this course have? Or I think the attendee means what kind of computer requirements does he or she need to have before the course? Anybody
Yeah,
that
I think
wants-
any
yeah.
modern computer and laptop would work because what we are trying to do is to utilize a lot of cloud resources and servers which are there to reduce the computational time or to the need of having in-house computational infrastructure, which is not possible. So learning about other options is also there. We're teaching that how to use cloud resources for such analysis when they're doing it independently. But yeah, throughout the course, any normal computer which are like post, I think 2016 would work fine, and also like other computers would work uh, fine as well.
Okay. Okay. So I think it's 4:15 and we would like to stop on time. So thank you so much, Premas Life Sciences, Shrishti, Rahul, and Omics Logic, Mohit, Subhangana, Praveen, Mitali, Srigori, everybody, and Team India Bioscience. We've had a lot of questions and we still have a few more. I think you will be spammed with emails now. And the last date to apply for this course is July 25th. So hoping that you'll also receive a lot of applications soon. So thank you so much
Yes, we
and
look forward to it.
Thank you, Devices.
Thank you very much.
thank
have a
you. Thank you
great
so
day
much.
ahead. Yeah.
Thank
Thank you
you
so
very much.
much.
Bye.
Bye.
Bye.